All right, welcome back to this chapter 6 video. We're going to talk about the ecological importance of microorganisms, specifically bacteria. And basically, without bacteria, life as we know it would not exist. We as humans would not be here on this planet if it weren't for these bacteria. They play a vital role in element cycles. So the carbon cycle, nitrogen cycle, sulfur and phosphorus cycles. We're going to look at the carbon cycle and explain why this is important for us as humans. In order to reproduce, organisms need to obtain or make nutrients. And one of the nutrients, like we mentioned in the chapter 5 video, is carbon. It's the fundamental element of all organic chemicals. So we're largely composed of carbon. And here, as you can see in this little amino acid structure, carbon is the backbone element. The start of the carbon cycle is autotrophy. So primary producers or autotrophs, they convert carbon dioxide and put that carbon dioxide into organic molecules. So they convert, they fix carbon into usable forms. Cyanobacteria, which are pictured over here on the left picture, are prokaryotic autotrophs, and algae and plants would be eukaryotic autotrophs. These are photosynthetic autotrophs. There are other types of autotrophy, but in the book we focus on the process of photosynthesis, which cyanobacteria, algae, and plants use. So cyanobacteria are the reason why we have an oxygenated planet. These organisms show up about 2.5 to 3 billion years ago, and they begin producing oxygen. So as a result of carbon fixation, these organisms produce oxygen as a product. So the planet basically during this time switched from an anaerobic to aerobic environment. And it didn't happen overnight. It actually took about 200 million years for there to be enough oxygen buildup in the atmosphere for aerobic life. Because what happened was these organisms, cyanobacteria, are aquatic organisms, and what happened is dissolved iron in the ocean captured that oxygen, and the oxidized iron fell to the ocean floor. So in figure 6.1 in your book, we show this basically it's a rock. It's a fossilized banded iron formation. This is evidence for this oxygenation period of the planet, which occurred for about 200 million years. So right there at the beginning of the carbon cycle, if it weren't for cyanobacteria, aerobic life might not have ever come about. So autotrophs give rise to consumers. Consumers are heterotrophs, and they utilize organic materials from other organisms. And we complete the carbon cycle by releasing carbon dioxide as a product of our respiration. So we incorporate that organic carbon that's made by autotrophs into tissues, and most of that carbon remains with us until we die. So we rely on the activities of those primary producers in one way, shape, or form. There are also decomposers, which are heterotrophic organisms, and they digest and utilize carbon from dead organisms. So bacteria and fungi play the major role in decomposition and release carbon dioxide as a product. So we have carbon fixation, we have metabolism, which completes that carbon cycle in a very simple way. The release of that carbon dioxide starts that carbon cycle over again. And worldwide, we have a rate of carbon dioxide production exceeding the rate in which it's actually being incorporated into organic materials. So think about why that may be the case. So why is there more carbon dioxide being put into the atmosphere than what's being taken out? And it has to deal with how you move around your neighborhoods and travel across the state and whatnot. It has to deal with fossil fuel consumption. So fossil fuels are fixed carbon and it's trapped carbon, and combustion returns that carbon back to the atmosphere. So this simplified carbon cycle showing the atmospheric carbon dioxide in the atmosphere and dissolved in water. Um, Carbon dioxide needs to be fixed, and you can see here there's only one arrow going out of that atmosphere or the dissolved CO2 in water. Respiration puts that carbon back into the atmosphere. Um, so we'll just leave it right at that. You can pause this video and further look at the follow the cycle and where the carbon is utilized. The nitrogen cycle, as we mentioned before, nitrogen is a growth limiting element. It's an important nutritional element required for proteins and nucleic acids. And most nitrogen is found on the planet as nitrogen gas. And nitrogen gas is not usable by organisms. So how do plants and animals get nitrogen? Well, we need nitrogen fixation from prokaryotes. So since nitrogen gas is not a usable form, consumers such as us, we must ingest plant and animal food in order to gain nitrogen. But we have to rely on nitrogen fixation from prokaryotes in order to do that. So nitrogen fixation, again, it's done by certain prokaryotic organisms, and only these prokaryotes can do this. You get nitrogen fixation through lightning strikes, but it would not be suitable to actually support life as we know it without these nitrogen fixing bacteria. So during nitrogen fixation, it's a process which nitrogen gas is reduced to form ammonia or NH3. Ammonia can be incorporated into cell material. So it's very energy taxing for the organism. It takes about 16 molecules of ATP for every molecule of nitrogen that's fixed. So organisms that are capable of doing this are called diazotrophs. And we can find these organisms either free living in soils and some aquatic environments or in a symbiotic relationship with plants. So if you're familiar with legumes at all, alfalfa, clover, peas, beans, peanuts, these organisms have a symbiotic relationship. They actually house the diazotrophs in root structures, and the bacteria fix nitrogen right for the plant, and they will release nitrogen into the soil as well. Ammonification is another process that bacteria will use if they're not capable of fixing nitrogen. So only a few types of organisms can do that. So bacteria and fungi, they decompose waste, so dead organisms, to produce and obtain nitrogen. So it's a decomposition process that converts organic nitrogen, and it's a harvesting of the amino groups from the protein. So here in the protein, this little amine group is recycled basically to make ammonia. So I have a picture of this chicken house here. If you've ever been inside a chicken coop, that decomposition of all that manure can result in lots of ammonia being produced. And what's important here is that ammonia can be converted to ammonium with a little bit of water. So organisms can also absorb ammonium. There's a process called nitrification. This is done by nitrifying organisms. They oxidize ammonia to nitrite, and then ultimately to nitrate. So nitrate is also another usable form by organisms. If you buy fertilizer for your garden or your lawn, that nitrogen, the NPK, the N is the nitrogen, that nitrogen is also often in a form of nitrate. And then denitrification is a process that converts nitrate back to nitrite back to nitrogen gas. So denitrification is completing that nitrogen cycle. So the nitrogen cycle begins with nitrogen fixation, and then it is ultimately completed by the denitrification process. So starting with fixation, ending with denitrification. So some organisms from the genus Pseudomonas are capable of this process. And it's an anaerobic process, and what's happening is the bacteria is actually using nitrate and nitrite as an electron acceptor in an anaerobic respiration process. In the lab, we will do a test to see if the organism can use nitrate as an electron acceptor. If you Google search not a video, a image for the nitrogen cycle, this is often one that comes up. The quality of this image is not very good. But again, we're beginning with nitrogen in the atmosphere, and we have nitrogen fixation by bacteria living in the root nodules of legumes, such as beans, soybeans, green beans that you grow in your garden, and then nitrogen fixation in done by soil bacteria. And then again, denitrification, returning that nitrogen back to the atmosphere. So we can apply this knowledge of understanding that organisms need these elements for growth, and we can apply it to fields in environmental microbiology. And one of those fields is bioremediation. So we're taking advantage of microbial adaptation and understanding that they need these elements for growth. And we can use those organisms to actually clean up toxic or hazardous pollutants, and the organisms will actually degrade them into harmless compounds. So the most known application would be using bacteria to clean oil spills. There's two types of bioremediation. There's natural bioremediation, or as we talk about it in the book, biostimulation. So you'd want to be familiar with biostimulation. I believe that's what's on your study guide as well. But what we're doing is we're encouraging the microbes to degrade the toxic substance. And we do that by basically fertilizing the environment. So we add the nutrients that are limiting to stimulate microbial growth. So in the case of an oil spill, oil is hydrocarbon, which has lots of carbon and basically no nitrogen. So you add nitrogen, you add phosphorus, you add potassium to that environment, and that's stimulating the microbes in the population to use the oil as a carbon source, and by providing the fertilizer, they have those other elements needed for growth. And then there's bioaugmentation, which you're adding the microbe that's not normally in the population. So sometimes that organism could actually be genetically modified to degrade a specific pollutant. So sources of pollutants, either domestic or industrial waste, like I mentioned before with oil, dioxins, herbicides, pesticides are all capable of being broken down by certain organisms. Now, synthetic compounds with similar chemical composition and structure are more likely to be degraded. Now, there are chemicals which are referred to as xenobiotics, which are synthetic compounds. These are totally different than what's naturally found in nature. So these xenobiotics are often persistent in nature for long periods of time. And they persist because organisms, they lack enzymes that are necessary for degrading them. So they lack enzymes that can actually recognize the chemical as a food source, basically. This figure is similar to what's found in your book. We have relative herbicide concentration. So herbicides are chemicals used to kill plants. So like weed killer, Roundup, for example. So we have an herbicide concentration over time. This herbicide that we're looking at is 2,4-D and then 2,4,5-T. 2,4-D is an herbicide you can spray on your lawn. It won't kill the grass. It'll kill weeds, though. And 2,4-D is readily degraded by microbes over a period of about 10 days. The chemical structure here is shown, and 2,4,5-T, everything is basically the same between 2,4-D and 2,4,5-T, except the addition of this chlorine here at the fifth carbon on this aromatic ring. All right, so one chlorine difference, and you can see here with this purple line, 2,4,5-T is a xenobiotic. It persists for long periods of time, so 25 days plus Who knows how long this will stay in the environment. Um, it is a persistent chemical. This picture is also found in your book. This shows the uh, beach shore after the Exxon Valdez oil spill. You can see here on the left the petroleum stained rock, and this is after a biostimulation, so fertilizing method to remove the oil. You can see it's not 100%, but it is a dramatic decrease of the hydrocarbon um, over a period of time. Biodegradation can be done in your backyard, so we can apply this to composting. If you want to decompose your kitchen waste, your vegetable scraps, food scraps, and things like that. Um, if you have the correct ratio of carbon and nitrogen, so your browns to your greens, add some water, you pile this stuff up. The microbial metabolism causes that pile to heat up to about 55 to 66 degrees Celsius, which kills pathogens at that temperature. Aerating them frequently can result in a rich organic fertilizer in about a 6-week timeline. So this is beneficial to help reduce landfill waste. This concludes the Chapter 6 video. Make sure you complete the Chapter 6 study guide and the Chapter 6 e-learning quiz.